Thank you to the organizers for offering me the chance to present my work. The title of my work is Metabolic Modeling in Human Milk Oligosaccharide, Biosynthesis, and Implications in Maternal Blood Groups. HMOs are lactose-derived multimeric, are lactose-derived multimeric sugars, which are a subset of the multimeric sugars that occupy and moderate up to 90% of the extracellular space. There's a growing understanding that HMOs are essential in communicating important environmental information from mothers to infants during nursing. As such, they are becoming a subject of greater and greater interest in the field of nutraceuticals. If nothing else, I hope you'll take away from my talk that HMOs have some impact on infant health, that they are able to be metabolic, they are suitable for metabolic modeling, and that that in this project we aim to resolve ambiguous gene reaction associations. So what exactly are HMOs? As I mentioned, they're lactose-derived molecules. Starting from lactose, you can take a small sugar, another sugar, add it on using a galactosyltransferase like FUT2, and create a slightly larger sugar. Now, this is a stoichiometrically balanced equation, and thus this system is amenable to metabolic modeling. Now, not only that, but this molecule, 2-fucosylactose, or 2FL, has been seen to prevent bacterial diarrhea. This is our first instance of the health protective properties. Now we can add many more sugars to this lactose to create larger molecules like DSLNT, which has been shown to prevent necrotizing enterocolitis in rats and has been shown to simultaneously be missing from milk fed to children who suffer from this disease. Now these are just two of the hundreds of observed and thousands of predicted human milk oligosaccharides that are believed to exist in the human milk. Now, how does this tie into blood types? Well, it turns out there is a heterogeneity in the FUT2 gene, which we discussed earlier, which is necessary for producing the 2-fucosylactose, which can prevent the production of blood antigens, resulting in what's called the non-secretor blood type. Non-secretor blood type, since it uses the same glycosyltransferases to build the blood sugars, the sugars that define blood types, can also reduce the diversity of milk sugars, which can have a health implication theoretically, not only theoretically, but in practice as well, as we see in the 15% increase in mortality in the NICU from children fed with non-secretor breast milk. So, in this project, I'm going to speed up a little bit because I'm running out of time. We want to provide the possibility of HMOs as a nutraceutical, as well as understand the milk types. That means we need to be able to synthesize them. That means we need to be able to reduce the possible genes that might be performing these reactions, which means we need to solve this difficult problem of assigning genes to reactions, which is currently an open problem. We also want to better understand the milk types by solving these biosynthetic models. These problems are intertwined, and I'm going to show you in the next slide how they fit together. So taking all the possible reactions that we could use to build these sugars, we're going to build them into a complete network, pare that network down based on the data that we observe, and then using that reduced network, we're going to pull out all the feasible models that can recapitulate the data successfully, and that's going to give us the activity. For those of you who are familiar with this field, flux variability analysis, MILP, FVA. Sorry for being curt. Then we're going to compare the gene expression to the flux values. And if you're interested in how I actually do this, it's too complicated for a 5-minute talk. Come to my poster, and I'd be more than happy to discuss it at length. My poster is 237. In conclusion, we were able to assign 8 of the 9 unknown GPRs, and 5 of those fit into the canonical lists. The 3 left over, we believe, are better answers than the canonical based on supporting information, which I can share with you in my poster. And we were able to locate the fucosylations, which we believe to be definitive of the secretor blood type and milk type. Thank you to my collaborators, particularly Anne Rochelle, who's my co-first author on this project. Thank you to ISMB and SysMod for providing me with the funding to come today. and thank you for your attention.